बिस्मिल्लाहिर्रहमानिर्रहीम। अस्सलाम वालेकुम डियर स्टूडेंट्स। आज बीएससी बॉटनी का चैप्टर दी फंजाई में से जो डिवीज़न हम पढ़ने जा रहे हैं वो है बेसिडियो माइकोटा। उसका बहुत ही इम्पोर्टेंट जीनस हम पढ़ने जा रहे हैं पक्सीनिया। सबसे पहले इसकी इंट्रोडक्शन हम देखते हैं। जो पक्सीनिया है ये इस जीनस की बहुत सारी स्पीशीज़ हैं। पक्सीनिया इज़ अ ओब्लिगेट पैरासाइट। ओब्लिगेट पैरासाइट का मतलब ये है कि दे डिपेंड अपॉन होस्ट फॉर सर्वाइवल एंड रिप्रोडक्शन। ये अपनी सर्वाइवल एंड रिप्रोडक्शन के लिए होस्ट पे डिपेंड करते हैं कम्प्लीटली। पक्सीनिया की एक स्पीशी है पक्सीनिया ग्रामिनस ट्राइटाइसी इज़ अ कॉमन स्पीशी ऑन व्हीट एंड आल्सो वेरियस स्पीशीज ऑफ ग्रासेस जैसे कि ओट बार्ले वगैरह में। जो पक्सीनिया है ये हेट्रोशियस है, यानी कि ये फंगस अपना लाइफ साइकिल कंप्लीट करती है लेकिन टू होस्ट में। एक होस्ट इसका वीट प्लांट है, दूसरा होस्ट बारबेरी प्लांट है, BARBARY बारबेरी प्लांट। सो लेट्स डिस्कस द लाइफ साइकिल ऑफ पक्सीनिया। It consists of 5 stages. The first is eurydinyl stage, number 2 teleal stage, number 3 basidial stage, number 4th pycenial stage, and number 5th axial stage. So now we are going to discuss first 3 stages in today's lecture. The first stage is eurydinyl stage. In eurydinyl stage, actually, for example, this is a stem of wheat plant and this is the epidermis of stem of wheat plant. In the stem of wheat plant, there is a stalk like structures are formed. You can see these are the stalks. These are the stalks are formed. Above the tip of stalks, structure formed named as sorai. And within each sorai, a number of spores are produced. Because this is a eurydinyl stage, so you can say uridosorai, and spores which are present in uridosorai are uridospores. And if you look at the structure of this is a uridosorai in blue in color. Uridosorai have uridospores. And uridospores, the characteristics of uridospores are they are binucleated, globose in shape, and thick-walled. This one is the uridospore. Binucleated, thick-walled, and globose, globe-like. So when they are released, these are all the uridospores, and this is the shape of uridospore. These uridospores fall on the surface of a leaf of wheat plant. When they fall on the surface of leaf plant, they undergo a process of germination. How? For example, this is a uridospore. Because uridospore are binucleated and uridospore form a germ tube on the surface of leaf. For example, this is a leaf and this is a stomata. This one is the stomata and this is the surface of leaf. Uridospore form a germ tube and germ tube at the tip of stomata swell. At the tip of stomata, germ tube swell, and both of these nuclei and cytoplasm migrate into this swelling named as aprisorium. Aprisorium. So these both nuclei move towards aprisorium and cytoplasm as well. And by a septum, by the formation of a septum, this gets separate from this uridospore, and two nuclei migrate into the swelling named as aprisorium. And these two nuclei fuse to each other and form a germ tube in the stomata, stomatal chamber. This aprisorium enter into the stomatal chamber. You can see by forming a germ tube, or you can say by forming a hyphae. These hyphae have a hook-like structure. For example, uh, these are the two nuclei fused, so these form a hyphae inside the stomatal chamber, and at the tip they form a hook-like structure, hostoria. And this hostoria help in food capturing. Hostoria hook-like structure. So that was the all about the urinary stage. Now next stage is teleal stage. In teleal stage, you can see this is the mycelium and this is the hyphae. From body of fungus, thread-like structures are arises named as hyphae. Above the tip of hyphae, a structure is produced, teleutosauri, and inside teleutosauri there are a number of spores called teleutospores. These teleutospores are dark red in color, binucleated, and they fall end of season. Is it clear? Now the third, this is the production of teleutospore, so we called second stage teleal stage. So now the next is basidial stage. These teleutospore fall end of the season. When these teleutospore fall, these are the teleutospore, they form a germ tube. They because they are binucleated, so their nuclei fuse to each other and form diploid zygote. And this is a germ tube. This is teleutospore which is now diploid in zygote. This zygote undergo meiosis and form four haploid nuclei. Four haploid nuclei and cytoplasm move into the germ tube, and you can see here 1, 2, 3, 4, and each separated by a septate, mean to say cross wall. In each cell form a structure named as stigmata, and now this stigmata by mitotically division form a chain of spores named as basidiospore. So due to the formation of basidiospore, they are called basidial stage. These are the nuclei. So today we have discussed the three stages: uridineal stage, teleal stage, basidial stage. Tomorrow we are going to discuss pycinial stage, axial stage. Thank you very much. Allah Hafiz.